Welcome to Web Microbiology, podcast for Bio 204 at Allegheny College of Maryland. I'm Professor Kelly. Let's get started. Episode 6, Guess Who? Welcome to Chapter 18, which is another chapter in your text that applies to lab. Even though you've only physically performed a few of them, you've already learned dozens of lab techniques for this course. So why have we been doing all these tests? The most relevant answer is it helps you understand the metabolic processes we'll discuss later in lecture. But also, clinical microbiologists use them to identify unknown microorganisms causing disease. Imagine that a patient arrives at the hospital with a serious infection. Symptoms alone rarely identify the exact pathogen responsible. Fever, inflammation, pain, coughing, diarrhea, wounds can all be caused by many different microbes. The lab becomes a detective agency, gathering evidence that will eventually reveal the identity of the culprit. This process actually begins at the bedside with specimen collection. Proper technique is critical. Contamination can delay diagnosis and complicate results. So knowing how the laboratory uses a specimen helps you guys, your healthcare workers, collect it correctly in the first place. Now, once the specimen arrives in the lab, microbiologists and lab technicians begin applying the same 5 I's that we've already discussed: inoculation, incubation, isolation, inspection, and identification. Depending on the infection, that specimen that was sent might be blood, urine, sputum, stool, could be a wound swab, cerebrospinal fluid, or any other kind of body sample. Now, I need to switch gears just a little because I think this is a great analogy. Do you remember or have your kids played the game called Guess Who? If not, I would look it up because phenotypic identification of bacteria is very much like that game. Now, instead of asking questions about hair color or glasses, the observable traits of people, microbiologists use the observable traits of bacteria, so things like cell shape, stain characteristics, growth patterns, and biochemical reactions to eliminate suspects. So each test provides another clue, narrowing the field until only a few likely or a single candidate remains. Clinical laboratories want to work efficiently and provide answers as quickly as possible, so identification really begins long before biochemical testing. The specimens are often inoculated onto multiple types of media. They're incubated under different atmospheric conditions all at the same time, so that by the time those colonies are isolated, we already have gathered a lot of valuable information about our unknown. Now, after inoculation and incubation, colony morphology can reveal many more important clues. Is the colony large or small? Is it smooth or rough? Colorless, or does it have a pigment? Does it produce hemolysis on blood agar plates? Does it grow in selective media? Does it grow under aerobic or anaerobic conditions, or both? Does it ferment lactose? These observations seem pretty simple, but they can eliminate huge numbers of possibilities, and here's the key. Every question I just asked can be answered using just your eyes, special culture media, and controlled growth conditions. So far, that microbiologist does not need a microscope. Now, after they do that gross inspection, then we move to the microscope. Now, in our game Guess Who, one of the most effective early questions is often, is your character a male or a female? Because it eliminates about half of the remaining possibilities. A gram stain is kind of a similar question for our unknown bacteria. Within a few minutes, this test can separate organisms into 2 major groups while also providing information from basic staining about cell shape and arrangement. A report of gram-positive cocci in clusters immediately suggests a different group of organisms than gram-negative rods, for example. These observations narrow the suspect list and help determine what tests those lab technicians do next. So we've got gram stain, colony appearance, growth characteristics, and also that patient history. All that information so far can reduce thousands of possible pathogens to only a handful. And that patient history contributes so much valuable information. This is another reason clinicians should understand how microbiologists do their work. So collecting a thorough patient history, including any recent travel, animal exposure, food exposure, environmental exposure, immune status, and medical procedures can all provide more clues. So lab identification really has to be integrated with the bigger clinical picture. Now at this point, the lab has kind of a working hypothesis and a group of possibilities. The one big problem, though, is that many bacteria look alike. So unlike the Guess Who game, bacteria only wear, if you will, so much of their phenotype on the outside. So the next step in identification is to perform these biochemical reaction tests. Biochemical tests are based on pretty simple principle. Different microorganisms possess different enzymes and do different metabolic pathways. They basically break down nutrients and things from their environment differently. So those differences create unique biochemical fingerprints, and by observing how an organism responds to specific tests, microbiologists gradually eliminate possibilities until only one or two likely identities are left. These are like asking the questions from Guess Who. Does your person have a mustache, wear a hat, or have brown hair? A biochemical test provides the answer to those questions in our bacterial Guess Who game. Now, if you're really good at Guess Who, the key to winning is to pick questions that eliminate the largest number of possibilities on each of your turns. Microbiologists and lab techs work basically the same way. For every question and answer, the next big or best question is developed. And in this way, the patient and the clinician both get the answer quickly. Now, although there are hundreds of biochemical tests, only about 10 or 20 of them are commonly used for identification purposes. And again, you'll have completed a lot of those in lab this semester. But a big difference is that the microbiologists don't perform every possible test for every sample. They choose only the tests that provide the most evidence to help them identify their unknown. Now, the identification process is often guided by dichotomous keys, which function like a series of branching yes or no questions. The next question depends on the answer to the previous one, and by following this formula, we narrow down the possibilities until that organism can be identified. Now, you'll practice doing just this in Lab 10, and I'll talk about that in the next episode. Biochemical testing is powerful because multiple results are interpreted together. But remember that up to this point, all the observations the lab has made on an unknown sample specifically evaluate phenotype, or the observable characteristics of bacteria only. We haven't yet looked at DNA or immune reactions. Phenotype identification centers around evaluating what that organism can do. Now, in the next episode, I'll explain how to work through the process of identifying an unknown sample using the same tools we talked about in this episode. I'll see you soon.